Hi everyone. In this video we're going to talk about osmosis. So osmosis refers to the diffusion of water across a membrane down its concentration gradient. One thing that's very important about water is that it actually moves opposite of the solute gradient, so we can also say that it moves towards the higher solute concentration. So let's break this definition down a little bit. Let's draw a cell membrane, and in the cell membrane we're going to draw an aquaporin. So you may remember from a previous video that aquaporins are needed in order to move water across the membrane. So aquaporins are channel proteins that are specific for the movement of water. We know that if water tries to move through the phospholipid bilayer, it would just bounce off. It's not lipid soluble. So if we want to move the water across the membrane, we can just watch the water molecules diffuse through the aquaporin. So the first part of the definition, diffusion of water across the membrane, it must be remembered that in order for the water to move across the membrane, there has to be aquaporins present. If there are no aquaporins, the abbreviation for aquaporins is AQP. If there are no aquaporins, that means that the membrane is not permeable to water. It's possible for a membrane to be permeable to other types of solutes and not be permeable to water. It's also possible to not be permeable to solutes and be permeable to water. But it's important to remember that in order for water to move, the aquaporins have to be present. So now let's consider the second part of the definition. So we'll draw the cell membrane again, and in this drawing we'll again add our aquaporin, and we won't add any other types of channels. And then we're going to draw a solute. And so we'll say this solute is, doesn't really matter what the solute is, but it's definitely not lipid soluble. So it's got to be either maybe glucose or sodium or potassium. It's going to be something that can't cross the membrane without the use of a channel or some other type of protein. And then we'll also draw in some water. The high solute concentration present on one side of the membrane opposite the low concentration of solute on the other side means that there is a solute gradient. Since we mentioned that the solute is not lipid soluble, it can't penetrate the membrane, so it's just going to bounce off. So the membrane is impermeable to this solute. Now, although it appears that the water concentration is equal on either side of the membrane, in comparison to the solute, we would say that there's a relative low water concentration on the side with the high solute and a relative high water concentration on the side with the low solute. So the water can cross the membrane. The membrane is permeable to water because of the presence of the aquaporin channel. And so water is going to move down the water gradient from an area of high water concentration to an area of low water concentration. So it's going to move through the channel towards the low water. Now, water also moves up the solute gradient. So water moves from the area of low solute concentration to the area of high solute concentration. So this is really the same thing. It's basically saying the same thing in two different ways, and it just sort of depends on how you look at it. But the reason why I do this is because it's very important to note that in cases where the membrane is permeable only to water and not to a particular solute, it will appear that the water is moving towards the high solute, and so it's necessary to specify when you say the water is moving, which direction it's moving, and whether that's based on the water gradient or the solute gradient, because they are opposite. So we can revisit our questions from last time. The first is, why does the water move? And the answer is to eliminate the gradient. So same as the reasons why the solute molecules move. And then, why does water need a channel? Because water is a polar molecule, so it can't pass through the phospholipid bilayer. All right, now let's take a look at another example in which we have a combination of both aquaporins and some other type of channel. So we'll draw a cell membrane with two aquaporins, and we'll draw in a sodium channel. And this sodium channel we'll say is open. So we won't specify what type of channel it is, we'll just say it's open. We'll draw in the ECF and the ICF, and we'll draw in some sodium ions. So we know that sodium is highly concentrated outside of the cell, so in relative comparison, more sodium ions in the ECF than the ICF, and then we'll draw in some water. And in this example, again, we'll draw the water to be in approximately equal amounts on either side. But because we have a high sodium concentration on the outside of the cell and a low sodium concentration on the inside of the cell, we have a relative low water concentration outside of the cell and a relative high water concentration inside of the cell. So because of this, we have a gradient. And the fact that we have a gradient means that we're going to have movement of stuff down concentration gradients. So we can actually show that since there's a sodium gradient and the sodium channel is open, the sodium can move through the sodium channel down its gradient, and the water, which is going to move in the opposite direction because it's moving towards the high solute concentration, can also move, and so it's moving down its respective gradient. So whenever we have multiple transporters present, we have to take into consideration the possibility of movement of multiple things, solutes and water. But I do want to remind you that this is a theoretical example. It doesn't take into account the full picture that would be associated with movement of stuff across a membrane. All right, so now let's draw a cell. And in this cell, we're going to show concentration of water and solutes both inside and outside the cell. And we're going to say that these concentrations are approximately equal. And we're going to draw into the membrane a bunch of aquaporins. And so we know that the cell is permeable to solute. And we're not drawing in any other types of channels. So we'll say that it's not permeable to any solutes at this time. So what I want to point out here is that the way we've drawn it, and we just kind of have to take my word for it, I guess, there's no gradient. There's an equal solute concentration inside of the cell and outside the cell, and an equal water concentration inside of the cell and outside the cell. So because there's an equal solute water ratio, there is no gradient. There's no difference. That doesn't mean, however, that there's no movement. If the channels are open for the water to move, then the water will move through. But in the same way we think of an equilibrium chemical reaction, we have an equilibrium situation where the water will move into the cell at an equal rate that it moves out of the cell. So it's moving, but it's moving evenly in both directions because it's in equilibrium. Now, if the circumstances were to change, then the behavior could change of the water as well. So now I'd like to introduce a concept called tonicity. Tonicity is basically used to compare the solute concentration of one solution relative to the solute concentration of another solution. Okay, so let's consider a blood vessel. And we know the blood vessel is going to contain a couple of things, including plasma and a red blood cell. So the red blood cell is actually the classic example when describing tonicity. And so we'll just sort of compare the concentration of solute and water inside of the red blood cell to the concentration of solute and water in the extracellular fluid. In this case, the ECF is intravascular. So in this drawing, the solute to water ratio inside of the cell is going to equal the solute to water ratio of the plasma. So we would say that the plasma is isotonic to the red blood cell, meaning it has the same solute to water ratio or the same solute concentration. Then let's consider the same cell in the same blood vessel, and we've established IV access. So we're going to administer some IV saline. And so saline comes in a variety of salt concentrations in the water, and we'll just sort of show a couple of examples for the sake of comparison. So we'll have our IV poked through the skin and into the blood vessel, and then we can administer whatever we want through our IV. So we're going to start with normal saline. So if we add volume to the vasculature in the form of 0.9% normal saline, 0.9% normal saline is isotonic to plasma, meaning it has the same solute to water ratio or the same solute concentration as plasma does. And so when we add this extra water and salt into the blood vessel, we'll have still movement of water into the red blood cell and out of the red blood cell, presuming that we have aquaporins, but we'll say that we do. So we'll have movement into the cell and out of the cell, but we have an equilibrium situation, so there's going to be no net movement of water. So let's compare that to another type of saline. So let's consider 3% saline. So we've administered 3% saline, and we can see that now there's a very high solute concentration in the plasma. We haven't seen any change yet in the cell, but we can see that there is a high concentration of solute in the plasma. So we describe the 3% saline as hypertonic to the plasma. It's more highly concentrated with salt particles, and so therefore it actually increases the concentration in the plasma. Now, because we have this increase in concentration in the plasma, again, we'll say that the membrane is permeable to water, not solute, and we're going to have movement of water in order to equilibrate this situation. So the direction of movement of water is going to be out of the cell. So we can't move any salts because we don't have any channels in the membrane to move the salts in our example here, but we can move the water, and the water can move from an area of high water concentration to low water concentration towards the area of higher solute concentration. So what happens is, as the water moves out of the cell will shrivel, and it becomes like a little raisin, and we actually call that process crenation. And then one more, let's compare to 0.45% saline. So let's say that now we've administered saline, which has half the concentration of salt that normal saline does, and so we can refer to that solution as hypotonic, and it would be hypotonic to the plasma because it has a lower salt concentration than plasma does. And so now we see relatively low number of solutes in the plasma in comparison to what's in the red blood cell. So again, we have net movement. We'll say permeable only to water, not to solute. We have net movement of water into the cell. And as the water moves in, the cell will swell, and then eventually it will burst. And if the cell bursts, obviously it dies. So because we have too much fluid movement into the cell doesn't have the ability to accommodate that, and eventually it will just rupture. So the thing to keep in mind is that normal saline is appropriate for patients because we don't want to have these changes occur to the cells. And the times when the other types of fluids might be administered in the hospital setting would be based upon patient pathology. So the patient is already sick for some reason, and then certain types of fluids might be needed to correct whatever the derangement might be. That concludes our overview on osmosis, and we'll sort of pick up this discussion when we talk a little bit about things like filtration and hydrostatic pressure and osmotic pressure and movement of the fluids through the vasculature, above and beyond just talking about the movement through the cells. So I hope you enjoyed it. Thanks for watching.